Okay, I see we're at the top of the hour, and there will still no doubt be people joining a bit later. This time is a bit different, so we do appreciate people taking note of the time change, and also so that we could have Jack, who's in Australia, join us. So welcome everyone to today's pesticide discussion. I'm Professor Andrea Rother. I'm head of the Environmental Health Division at the University of Cape Town in South Africa. You are all most welcome. Do take a moment to indicate your stakeholder group that you represent and introduce yourself in the chat with your name, organization, and country. So the University of Cape Town's Pesticide Discussion Forum is supported by funding from Sweden through the Swedish Chemicals Agency, Chemi. And today I will be facilitating, and in the background, Dr. Natasha Lalou will be providing technical and administrative support. And just for those of you who've just joined us, please do continue to introduce yourselves in the chat. So the purpose of our discussion forum is really to be a community of practice of those of us who are working in the field of pesticides. And it's not a webinar, but really to foster a discussion. And the idea is to bring representatives from different regions, sectors, and stakeholder groups together to foster a mutually supporting consortium of stakeholders to discuss various issues related to pesticides, and often to bring to the table issues that are difficult for low and middle income country regulators and issues that are new, like today's discussion on RNAi pesticides. And the focus is really to build the capacity of regulators, researchers, and NGOs in low and middle income countries, where we hope to learn from each other, share information and data, identify gaps, and problem solve to reduce and prevent pesticide poisoning, chronic effects, environmental contamination, really with a view to a toxic-free future for the next generation. So this is a non-partisan discussion, which means we may not agree with each other, but we respect each other's viewpoints. When you do comment or post a question in the chat, it always helps if you indicate the country or organization you're from. And we encourage you to engage with each other's responses as well. We will obviously be posting questions and polls after the presentations, but we do encourage you to also stimulate discussion. So we have two esteemed presenters today that we are very fortunate to have to take us through this discussion on RNA interference-based pesticides. So really looking at the issues related to genes silencing pesticides, their benefits and concerns. So Jack and Magda are going to take us through the three questions. And next slide, please. I'm going to invite Magda to start, Magda from the OECD, and Jack, who's from the University of Canterbury in Australia. Magda, I'm going to hand over to you and I ask you to introduce yourself. Thank you very much for taking time to speak to us today, and you're most welcome.
Thank you very much, Andrea. Thank you very much also for the opportunity to be here. It's a very exciting and creative forum that you created. So my name is Magdalini Sachana. I'm with the OECD Secretariat. I've been working at the OECD the last 9 years. And between other things, I'm also coordinating the work on biopesticides and also the one on novel technologies, including RNAi. So before joining the OECD, I was an academic, a researcher, and I'm a veterinarian by training with a master's degree in biotechnology and a PhD in toxicology. So I will start by providing some historical context, explaining what made us start working on RNAi-based pesticides, and then we will dive more into science, but also into the regulatory aspects. So this is very introductory, the first presentation. So let's get started. First slide, please. Yeah, thank you. First, to explain a little bit about the OECD, for those of you who are not aware what OECD stands for. OECD stands for the Organization for Economic Cooperation and Development. So it is a forum where regulators from 38 member countries and the EU are gathering and discuss with stakeholders. This can be industry, but also NGOs, environmental NGOs, and animal welfare NGOs. Within this forum, we coordinate and harmonize policies, we discuss issues of mutual concern, and work together to respond to international problems. We also, within our remit, is to evaluate the readiness of emerging science and technologies for regulatory applications. So next slide. Okay, so in the OECD, we have a big program on chemical safety, and under the umbrella of this program, we have dedicated work on pesticides, where we develop practical and harmonized tools that countries can use to implement the legislations regarding the safe use of pesticides. I would imagine that you are aware of the OECD test guidelines. These are important tools for regulators to assess and test the safety of the chemicals. And when they are conducted under good laboratory practices, they're accepted by other OECD member countries, but also countries that adhere to the mutual acceptance of data. Furthermore, we develop electronic tools for data submissions and database. Some of them are the iEuclid, if you are aware of, QSAR Toolbox, Econ Portal, etc. And we have risk assessment methodologies, both for hazard and exposure assessment. So within this program, we focus also on sustainable approaches towards plant protection. So new technologies are core there for the design and application of pesticides, which reduce risk to humans and wildlife. Next slide, please. So I would imagine you're aware that there is this increasing concern about the use of chemical pesticides due to their toxicity. Some of them appear to be persistent in the environment, and others can have off-target effects to beneficial organisms such as pollinators. Or we lately see also an increase in incidence of resistance of pests to some of these the compounds. That's why industry, but also researchers started to working on alternatives pest control strategies, such as the use of small ribonucleic acid. These are RNA molecules that target only a specific gene in a specific pest. So we're talking about a very targeted pest control strategy here. And this is done through a mechanism that is known as RNA interference, or RNAi. Next slide, please. So what we are talking here and what we are covering in this webinar is this technology, but the application is totally different from the transgenic approach. So we are not covering this in-plant approach. We're covering this technology when it's used to spray as a sprayer, as a root for root delivery, for stem injections, or in bites and traps. Next slide, please. In a sense, we're looking to this externally applied double-stranded RNA-based pesticides, and what the regulators wanted to hear when this technology came out was, so if the current approaches that we have to environmental and human health risk assessment for conventional and biological pesticides, but also the knowledge that we have for regulating genetically modified technologies can be used to assess the safety of these products. Furthermore, the regulators wanted to see if there is need for any additional data requirements or new sets of data that may be needed for these particular products. That's why back in 2015, we established an ad hoc expert group on RNAi-based pesticides to work and address all these questions that regulators had for this new technology. Next slide. And the best way to start was by gathering in the same room the scientists, the regulators, industry, NGOs, and discuss all the issues that are related to this externally application of double-stranded RNA-based pesticides. So there was really focus in this conference on regulatory needs and aspects of risk assessment for these products. And there was some preparation. We had the draft document on the environmental risk assessment that then became our consideration document. That really paved the way to the discussion that took place almost 5 years back then. Next slide. This conference also helped to prioritize the issues on which this expert group on RNAi should work. And priority was given to the environmental risk assessment because also from the experience that was gained from assessing these technologies in genetically modified plants, there was some substantial knowledge for human health risk assessment, but not that much for environmental risk assessment. And there were some questions to be asked in relation to the environmental fate of these products and also to the effects on non-target organisms. For the human health, there were other questions related to the different routes of exposure, but if we're also protected due to these physical and biochemical barriers that we may have, we are equipped with. So these are considerations that I will come back in session 3 when we'll discuss. We'll dive much into the details of these issues. So next slide. So all this work allowed us to create a kind of one-stop shop. So we have a dedicated webpage, so you can find all the information about the conference that we held back in 2019. And if you want to really see a kind of snapshot of what was discussed and what was presented, I would recommend that you read first the conference report that was published. And then if you need more information about the science and all the regulatory considerations, I would recommend that you go to this special issue in the Frontiers in Plant Science, where the presenters subsequently to the conference really produced some really detailed manuscripts, so you can find lots of information there. Furthermore, if you're regulators and you want to really dive into considerations that you need to to be aware as you assess similar products, I would recommend these two consideration documents that we released, one in 2020, and this was on environmental risk assessment, and the second one last summer that is about the human health risk assessment. So next slide, please. So all this work that we've done at the OECD really paved the way to have the first registration of a topically applied double-stranded RNA product for control of Colorado potato beetle that happened last year in December. And through this press release from the USCPA, you can see that the OECD work is acknowledged, and maybe for me it's time also to acknowledge that this is OECD work, but we really rely on our member countries. So this effort was led by the US, but many other countries contributed. So we had experts from Canada, Germany, and also Italy. And technology, as I've said, scientists really contributed to this knowledge, and industry provided data so regulators can familiarize themselves with data and produce all these documents. So I think that's it for me now, and I'm happy to take any questions.
Thank you very much, Magda. You really gave us a good overview of what RNAi pesticides are, why they've come onto the market now, and also what the initiatives have been so far by the OECD and the US in terms of starting to think about risk assessment and regulating. And so it's been really useful. Thank you for that. And I'm hoping that our members will have many questions for you. So we have, what we want to do for this first round is ask you, what are your thoughts about RNAi-based pesticides? You know, is this kind of the first time you're hearing about it? What is your thought? What you've just heard from what Magda said? Do you think that they're a good substitute for chemical pesticides to tackle some of the issues that Magda talked about, like resistance, for example? So please post your questions in the chat so that Magda can respond to them. And we will also have a few poll questions. And if you have some experience with these pesticides, you can also post those in the chat for us to stimulate the discussion. I think one of the questions in looking at the paper that you circulated, Magda, was about, you know, at the moment, is there a good understanding about the potential health impacts? So you mentioned that RNAi pesticides are targeted control, and they target a specific gene in a pest. And is there any implication that there could be a crossover to targeting the genes in humans that are being exposed? And it's fine if you don't know the response to this.
No, that's fine. I mean, okay. I think this is something that we'll address in our final presentation, but I'm happy to take it here also. So, I mean, we have a historical knowledge about the safety of the naked double-stranded RNA. So we are exposed through our food to these molecules. So this is, you know, it's well known, it is well understood. We also have lots of barriers that can really protect us. So we have the saliva, the acidic environment in the stomach, all the enzymes, nucleases in the gastrointestinal tract that can protect us, and many other mechanisms. So we know all that, but and also we have a lot of scientific knowledge, because this technology has used in the field of therapeutics. So many of these double-stranded RNAs are used in for medicinal products, but to go to the target that they have to go, they need to be modified, chemically modified, or sometimes used in a combination of some conformulants. And this is what makes these molecules basically to cross some barriers. And these are the questions basically that the regulators have. And we'll discuss later on that so the evaluation of a product is shifted not only for the active ingredient, but also to understand these chemical modifications or coformulants, and assess these products as a whole, because that's important. If like a barrier is crossed, then we need to know if there are any adverse effects for human health.
Thank you. Thanks for that. And I didn't mean to preempt what's coming, just to get the conversation going.
No, we will go faster in the session.
No. Oh, right. So you can see on the screen that people are responding to the poll. So have you ever heard of these RNAi-based pesticides before this discussion? And of the 13 people who've responded, 7 have said that they haven't heard of it. There's also discussion going on in the chat. So there have been a couple of questions. I think Jack is responding to the one, which has been... maybe I'll, so it was this question around what are human risk assessments once the RNAi pesticide is stabilized. So Jack, I think you've been responding to that. Magda, the other question has been, let me see if I can read this. The limitations challenging greater adoption of RNAi-based biopesticides includes certain factors which influence efficacy by the nature of biological processes which underpin their activity. Effectiveness may be limited by the speed and persistence of pesticidal action, as well as the significance of timing applications with respect to target pest life cycle and population dynamics. How to address the efficacy challenges is the question.
Okay, I mean, this is, I think it's a very good question, but also to highlight that at the OECD, we're not really much working on efficacy aspects. There are other international organizations working on that. But as I've said, there is the technology there that makes these products more stable in the environment, and I understand that the scientists, the researchers, really diving to enhance their stability and also the efficiency to control pest diseases. So that's what I understand. I'm sorry, I have limited knowledge in this area. But for sure, I mean, it seems that they're picking up. They're coming more and more countries are starting to receive now applications on these products. But I mean, that remains to be seen how they're going to be used, if they're going to be used as standalone or in combination with some other pesticides. They may well be used, as a first, as a first year, and then followed by chemical application. That remains to be seen.
Thank you. I'm not sure if you're seeing the other questions. So from Robert, there's a question, are the current formulations designed to minimize drift that remain within points of application? So do they minimize drift? Sorry.
I mean, as I've indicated, there are different ways of administering this. Of course, if are sprayable, depending on the technology that is used to apply a sprayable product, you can reduce drift, but also there are other types of applications, as of demonstrating by injecting the stem, by route delivery, you can use bites. It depends on what you're doing, but of course, application techniques are improving, and they are becoming more also kind of focused and precise. So we expect that this will be applied also for these pesticides.
Thank you. So I'm going to give you one more question, and then we're going to move on to Jack. You can see on the screen also that, interestingly, people are responding that RNAi-based pesticides should be regulated like biological or genetically modified organism rather than conventional chemical pesticides. And that's maybe something that we can pick up a bit later on in the discussion. So the question is from Joan. Can you speak on whether there were in vivo experiments to understand or analyze off-target effects of RNAi spray, or just mathematical modeling? And there's a second part to that question, but I'll let you answer that when Jack's presenting.
That's a great question, and also this is something that we were planning to address in our last presentation. What the experts concluded in these documents that I presented earlier is that bioinformatics are valuable. They can give enough information, but they have to be also accompanied with some testing. So these bioinformatics can help us to identify, which other phylogenetic similar insects should be tested. So to make sure that indeed there is no dangers, there is no any effect in other insects, or even other phylogenetically distant insects, like for example, beneficial insects, pollinators, etc. So the recommendation from the experts was, bioinformatics sequence data is viable, but it is not enough. It should be used to dictate or understand which individual, which species should be tested, basically. And we'll come back to this issue later on also.
Thank you. Thanks very much, Magda. And thanks again for getting us going. There are a lot of questions and comments in the chat. So I'm going to ask if you could go through those and perhaps give some typed responses. Even Jones had a second component to it. And we're going to move on. And hopefully we'll come back to this poll a bit later. But Jack, I invite you now. There you are. So welcome, Jack. And thank you also for responding to some of the comments in the chat already. I'm going to hand over to you if you can introduce yourself, and thank you again for taking the time to be with us today.
Kia ora, hello everyone. I just need to correct something that was said earlier. So as much as I would like to be in Australia, I'm at the University of Canterbury in New Zealand. So it's a slightly different, little bit further over.
My apologies.
No worries. I just worry that you insulted the Australians. So it's a pleasure to be here. Thank you very much for having me. My name's Jack Heinemann. I'm a professor of molecular biology and genetics at the University of Canterbury in the School of Biological Sciences, and I'm a director of a small research center called the Center for Integrated Research in Biosafety. And I have been for a number of years, although not any longer, a part of the Cartagena Protocol Ad Hoc Technical Expert Group on biosafety, as well as having been an advisor to a number of different governments on various biosafety issues over the years. My task with you tonight, my first task with you tonight, is to talk about some of the more technical elements of these types of pesticides. And they're known by various names like genetic pesticides, double-stranded RNA pesticides, et cetera. And hopefully, as I go through this presentation, it'll become more evident to you how they get these various different names. Could I have the next slide, please? The key active ingredient that we're talking about is a molecule of RNA, but it's a double-stranded RNA molecule, and that property, that structure gives the molecule properties that we don't tend to associate with single-stranded RNA. So over the many decades where we've been fixated on messenger RNA, the RNA that's used as the intermediate to produce proteins, that fixation has led to a great deal of knowledge and frustration about just how unstable RNA is. But those observations don't translate to double-stranded RNAs. So double-stranded RNAs are remarkably stable, far more stable than single-stranded RNA. And in some cases, double-stranded RNA is also more stable than DNA. So while there are enzymes that will degrade double-stranded RNA, it has become known now to be surprisingly a robust molecule. What I want to show you in this slide, in particular on the left-hand side, are the types of structures that we refer to as double-stranded RNA. They can come about by two different RNA molecules that have a sequence that allows them to base pair with near-perfect complementarity, or the double-stranded component of this molecule can form spontaneously by a fold in the RNA molecule where two different domains are complementary and will form base pairs of near-perfect complementarity. That second structure, shown at the bottom left, is a stem loop structure. You can deliver double-stranded RNAs either as some version of a stem loop structure or as two different molecules that form intermolecular base pairs, depending upon what your strategy is. What is shown here on the right is the RNAi, the RNA interference biochemistry that we know of from eukaryotic organisms. So that'll be the plants, animals, and fungi that are the targets of the pesticides we're talking about tonight. Now, I'm not going to go through all the biochemical steps, but what I want to illustrate in this sequence is that a number of events come together, and those events are also places at which we lose a little bit of track on the potential events that come about by using these RNA molecules. So the products of these RNA molecules can, in some organisms at some frequency, prime the production of more RNA molecules that lead to secondary and sometimes tertiary derivatives, which may not share a sequence similarity with the original formulation that we exposed an organism to. Next slide, please. RNA interference has at least three manifestations. Two of them are what we call the gene silencing that stops translation. So it's a post-transcriptional gene silencing. In post-transcriptional gene silencing, and you can see that pattern in the bottom right, the RNA works in the cytoplasm, finds a target RNA molecule, hopefully the target you intended for it, and it can either cause a translation inhibition, preventing a ribosome from binding and initiating translation on that messenger RNA. Or it may lead to a chemical modification of the RNA that increases its likelihood to be degraded through normal RNA degradation pathways. Or it might result in cleavage into ever additional smaller pieces of RNA, which also then can go on to prime the production of secondary and tertiary. Very often when people talk about RNA interference, they're referring to that only to that last pathway. But I think it's important to note that there are other pathways involved in RNA interference that aren't mutually exclusive. Just because one pathway is activated most of the time doesn't mean the other pathways don't happen as well. And in some organisms, it may only be the secondary pathways that are initiated from exposure. The third type of RNA interference occurs before transcription. So those same double-stranded RNA molecules can be transported to the nucleus, where they cause a series of reactions that result in chemical modifications of a DNA sequence with a high degree of similarity to the double-stranded RNA molecule. And that's shown in the lower left of this slide in the yellow box. Once that process finishes, those chemical modifications can then be inherited through cell division and sometimes also through organismal reproduction, through a process where when the DNA replicates, the pattern of chemical modifications that was inspired by the double-stranded RNA are preserved. So we have at least two different areas in which RNA interference can act. It can act on the DNA version of a gene, decreasing its rate of transcription or maybe even eliminating transcription. And it can act in the cytoplasm, causing a series of events with the produced messenger RNA, some of which are very strong, like that endonucleolytic activity, and some of which are not as strong, such as translation inhibition. Next slide, please. What we know is that, for the most part, RNA interference causes the silencing of genes, but it can also sometimes activate genes, particularly if it silences something that was already silencing something else. There are various pathways. Not all of these pathways are equally powerful in all organisms. They can vary between species. There can be differences, for example, in heritability, because some species have an RNA-dependent RNA polymerase and maybe some don't. We can't usually be confident that if we see an effect in one species of a lineage, that it will occur in the same way at the same frequency in other species of a lineage. But the effect can be long-lived. So once you create an RNAi effect, that effect can last for the lifetime of the organism. And for some organisms, like trees, that can be a long time. And in some organisms, particularly things like worms, that effect can be inherited for so far hundreds of generations. The next slide, please. Now, originally, originally double-stranded RNAs as a pesticide were delivered by making a plant transgenic. So this is an example of an early version where the corn has been genetically modified to produce a double-stranded RNA, which, when it's consumed by a pest of the corn, is converted into a toxic form of double-stranded RNA, toxic for that pest. Next slide, please. But as Magda said, the emphasis tonight is on new developments where the double-stranded RNA is delivered directly in the environment. Now, there's some advantages to this because it's scalable, just as chemical pesticides are scalable. But there are some disadvantages because you can't control the exposure as effectively as if you have a plant inside of a laboratory. And you can't, even for your target, but you can't control what other species, unintended target species who are not your target, and the potential for them also to react to your double-stranded RNA. So what's developed are these generally formulations that allow the double-stranded RNA to be encapsulated in a form that stabilizes it further and then assists it in getting inside the cells of, say, a plant or an animal. If your pest is a plant, then it's a herbicide. And if your pest is an animal, then it's a pesticide, an insecticide. So this is showing, for example, an idea that's based on countering Roundup-resistant weeds. So the weeds who have become resistant to the overuse of glyphosate, they can be made susceptible again to glyphosate by silencing the genes that make the plant resistant. And then you can go back and use a chemical-based herbicide to kill your plant. Next slide, please. So how does this work? Well, there's a variety of different chemical solutions to this problem. What's shown in this slide on the top and the bottom left are nanoparticles. So these are nanoparticles that will engulf the RNA, protect it in some form, and assist its transfer into the cell of an organism. You can also use hybrid nanoparticles and even viruses or viral bodies to help transfer this RNA. But for the most part, many of the formulations, many of the chemical adjuvants that we find in the formulations for small chemistry-based pesticides will also work with double-stranded RNA. And it also means that there's the potential for the capture of these double-stranded RNAs in existing formulations. But they don't have to be all that mysterious. Some of them sound scary, like organosilicons, but they're very common, and organosilicons are very good at getting nucleic acids inside cells, which make them very potent, whether or not the exposed organism was your intended exposed organism. But for double-stranded RNA, even sometimes just an abrasion effect will cause the RNA to be taken up. And water can be sufficient as a vector. So you can introduce the RNA through root absorption just by soaking the plant, or in the case of some animals like worms, soaking them in just an aqueous solution that has the double-stranded RNA. Next slide, please. And what makes this different now is that because of these delivery chemistries, the way delivery has changed, we can go into the environment and apply these pesticides at a greater scale. And that's what's made them commercially viable. So what's new? Well, what's new is that we get mass application. We can use mechanized means to deliver. This is sprays from tractors, drones, airplanes, which also increase the range of exposure pathways, taking them away from just ingestion to increasing contact or inhalation. There's less training required for the production and also the use of these RNAs than they were when they were pesticides in transgenic plants. And you can combine different active ingredients in different formulations to create ever more complex and adaptive forms of these pesticides once they're approved. Right. So that was just my little introduction to the technology behind the pesticides.
Thank you very much, Jack. You've presented a complex concept and process very succinctly and generated questions. So what questions do you have? We're asking now to the participants in relation to RNAi-based pesticides and or to assessing their risks. So there are a few questions already in the chat. I don't know if you can see those. Also, interestingly enough, you kind of stimulated the name aspect. I've noticed in some of the documents and publications you posted, it was more dsRNA, so for double-stranded, or Si for single-stranded. Which is the more commonly used of the three?
No, sorry, those are synonyms. So siRNAs still form double-stranded active ingredients. This is they're usually formed. Si usually means that they've been produced by someone and introduced exogenously.
Okay.
Whereas microRNAs, which are also double-stranded RNAs, are ones that are already produced by the genome of the organism. But in all cases, the instigating molecule is a double-stranded RNA molecule. Later on, very late in the process, through the targeting process, it becomes a single-stranded molecule, but the instigating form is always double-stranded.
So then referring to them as RNAi pesticides is going to be the more formal way of going forward. Okay.
Yes, because RNAi is the intended effect of all of these, no matter what nomenclature you used for your active ingredient.
Great, thank you.
I would just point out also that RNAi may be the intended outcome, but RNA has lots of roles in a cell. Small RNAs can have lots of roles in a cell. So there can be other effects of these, but one of the usually strongest phenotypically is RNA interference. And even though we don't have the same biochemical pathways in bacteria, prokaryotes, that cause RNA interference, small double-stranded RNAs are also gene regulators in prokaryotes.
So there's a question from Tom about what are the risks of resistance to RNAi pesticides?
Well, I imagine that the risk of resistance would be a commercial risk for the first part, just as the risks of resistance to glyphosate-based pesticides start to undermine the commercial model for those. There could be, I guess, more detailed pathways by which resistance could be a problem for us on a biosafety level. It might be that the mechanism of resistance creates a general resistance to inducing an RNA interference, and that might then result in a loss of an entire class of double-stranded active because of someone's use of it in one application.
So another question. So Christian from Tanzania has 3 questions. I don't know if you can see them in the chat.
Oh, I don't. I have the chat open, but I'm not sure I know which question you're talking about.
I can read. So are RNAi-based pesticides effective on weed and fungal control? I think you've already responded somewhat to the human health and non-target organisms, but the second part of the question is, how are the approval assessments done regarding approval of these products, and especially in low and middle income countries where there's not a lot of capacity?
All right. There are under commercial and research development double-stranded RNA approaches for fungi and as herbicides. I mean, I showed you BioDirect in my talk. BioDirect is a way to try to, again, control a weed. But RNAi-based pesticides are not common yet, right? So we have a couple of transgenic plants that deliver our double-stranded RNA pesticides. And we have now, like Calantha, recently approved, but is only just entering the market. So there's very little experience in the environment or for commercial providers for how this is going to turn out. I can say that, for example, in New Zealand, there's a trial where double-stranded RNA is being used to try to control the varroa mite in honeybees. But again, this is not, as far as I'm aware, commercially available. So the answer is we don't really know yet what will come down through the pipeline for these other kinds of pests. But in theory, there's no reason why a wide range of different species, including things as different as plants, fungi, and animals, could be targets for such pesticides. The second question was something along the lines of, how do you find out that it's safe? Well, for me, that's where we're at. We have, as Magda told you, the OECD got together and looked at this. We also know that the US FIFRA got together a few years before that and tried to look at this. The US National Academies commented on this. And the bottom line for me is that when you have an uncontrollable exposure, that is, you don't know who is going to be exposed to your spray, and you have a site-directed mechanism such as RNAi. I mean, everybody talks about the advantages of that site-directed mechanism, improving efficacy, but the extrapolation to safety is one that I don't make so lightly or easily, because the site-directed mechanism doesn't have to be, doesn't depend upon a perfect alignment between these molecules. But we also know that the base pairing between these molecules can be affected by all sorts of environmental conditions, such as drought or flooding, changing ionic concentrations. And therefore, it's at the moment impossible to predict all the unintended effects in unintendedly exposed organisms. So in non-target organisms, we can't predict all the effects, and we can't even predict all the effects in target organisms. So as Magda said, we can use bioinformatics to at least get a good guess at who is really likely to be affected. But we can't use bioinformatics to reassure ourselves that those are the only candidates for off-target effects. And since we know so very little about the biodiversity of the fungi, the useful, happy, necessary fungi in our environment, or the prokaryotes, it would be very difficult to assess the effects on those kinds of organisms using the tools that we have right now.
I think your last statement is the very good point about, in terms of the tools we have now. There's a question from Debbie, whether the claim is correct that RNAi pesticides are biopesticides. And this takes us back to the poll we had earlier, where we asked people to vote on whether they should be considered, as conventional bio, GMO, or something else, and that no one said they should be like conventional. 8 had said biopesticides, 10 GMOs, and 3 something else. So your thoughts on that?
My thoughts, well, first off, we do see different governments making different decisions. So one of the resource documents I gave you chronicles decision-making pathway in New Zealand, where initially the regulation of it was going down a pathway of a chemical. So they were going to treat the risks of a double-stranded RNA mainly based on the chemical RNA. And I think there are important shortcomings to that point of view. It has a molecule, an organic molecule. RNA is an active ingredient, but except in very rare cases, the RNA is likely to be delivered in a formulation, some kind of chemical formulation. And we can't ignore the fact that there will be a mixture of chemicals, some of which are the RNA, and some of which we wouldn't normally classify as, I guess, quote unquote, biological. So, and there may be, in cases, differences between this and the delivery package, which is a transgenic organism designed to produce the double-stranded RNA itself, with a pathway, say, of ingestion by a plant pest. So it really fits between a number of different categories, and I won't be surprised to see governments separating on this issue, depending on which regulatory pathway is going to optimize their goals of human health and environmental safety.
Thanks for that. There are quite a few questions. I think, Ahmed, your questions about, how do they function and how do they reduce gene expression? I think Jack presented quite well in his slides, and we will be sharing the slides with everyone. And there was a comment about, having the ability to test and assess. And as you were saying, some of the methods available at the moment is to be seen whether they work for these for the RNAi products. But the last question I'm going to give to you, and you can comment on the poll if you'd like as well before we move on to the third question, is from Nakati Singh, who's asking, are RNAi pesticides affordable and easy accessible to low and middle income countries?
Yeah. Once again, the commercialization of these is very new. So I really have no idea whether they'll be affordable. What I do think, though, is that particularly if you live in a biodiverse country, you may find it more difficult, more challenging to do the kind of risk assessment that you think is appropriate for your environment. And that, I think, will be a burden for low and middle income countries.
Thanks for that. So there are more questions to you, but we are going to move on to the third one. I don't know if you want to respond to this poll question. And then I will try and bring in some of the other questions when we're in question 3.
Do you want me to reply to the poll question?
Is there anything you want to comment about it? Is there anything in the
No,
responses?
I think it's a good segue for the next section. So I'll leave it at that.
Okay. All right. I believe there's a third poll, so go ahead and post that. And I'm going to actually then give you one more question while people are responding to the third poll. There are many, so I'm going to see. So there's a question from Claire. How can the off-target risks to human, animal, and environmental health possibly be tested or assessed, as they seem to have so many potential off-targets?
I think it is difficult, and probably the best chance of being able to test them, ironically, is in humans, but that doesn't mean that it is satisfactory for everyone what we can do in humans. The reason I say it's most likely to be easiest to test is because we know we can at least require those kinds of experiments, but it may be very difficult to get experiments done on an important organism in your country if it's not an important organism in the country of the manufacturer or the manufacturer's major markets. So I would think that for those who have an interest, particularly in environmental risk assessment, that the risk assessments that they will receive are ones that will be tuned to important species, off-target species in other countries, which may overlap with yours, but may not be a perfect recapitulation of your own environmental concerns.
So just a quick kind of follow-on question from David Kapandula, who was the regulator of pesticides in Zambia. Says, why then should we commercialize a technology whose environment and human health effects are not fully known, especially in low and middle income countries? So is this, you said that at the moment it's only being considered in high income countries. Why should low income countries maybe contemplate regulating these?
No, I didn't say that it's only being considered in high income countries. I don't know what countries are considering it or not. I just know that Calantha comes from a high income country. But I think that's an excellent question and not one for a molecular biologist. Other than what I would say is that if you want to pursue a chemically intensive agroecosystem, and that means using pesticides, well, then there may be a market for yet another pesticide to add to your chemically intensive agricultural model. It may not be the best solution or your only solution to growing food and or whatever other applications you have for pesticides in your country.
Thank you. Thanks for that. So I think this is a segue now into the final question that we're going to look at in the final section. So Jack and Magda, you're both going to present a bit on this. Magda, maybe you want to come back? And I hand over to the two of you.
Natasha, did you make a decision who would go first?
Yes, Jack, you will go first.
Oh, I'm going first. Okay.
No,
All
no
right.
pause for you.
All right, great. So I just have a couple of issues here to raise when we're thinking about doing an assessment of a potential double-stranded RNA-based, RNAi intended product. Next slide, please. You likely will frequently hear such statements that RNA is both ubiquitous and therefore safe. But remember, these double-stranded RNAs are designed to be toxic. So RNA is ubiquitous, and outside of a cell, RNA, as far as we know, is a safe molecule. But it is a bioactive molecule. And in the right structure and the right sequence, it can have a potent physiological and also sometimes heritable effect. You may hear that RNA is unstable and degraded, both inside, say, humans and in the environment. But a lot of this comes from our experience with single-stranded RNA, and double-stranded RNA is much more stable. But furthermore, when it's used as a commercial product, it's rarely going to be used as a so-called naked molecule of double-stranded RNA. It will be encased in adjuvants that further stabilize that molecule. And in many cases, there will also be a chemical modification of the component nucleotides themselves. And those chemical modifications are also intended to increase its stability. And finally, there are many adjuvants already approved for other pesticides that can be used also for double-stranded RNA. There may be a temptation to say, well, since we've studied that adjuvant in the context of a small chemical pesticide and the adjuvant itself doesn't raise any safety concerns, there may be a temptation to say that in the new combination with a double-stranded RNA, it doesn't raise safety concerns. And that has to be a decision for the regulator. But my advice would be that these are novel combinations in novel contexts. And it's also then possible that because RNA is ubiquitous, including small double-stranded RNAs, that the spray of the adjuvant may also be able to mobilize double-stranded RNAs that previously we were protected from and convert them into vectors for these other double-stranded RNAs. So it may be also a new pathway of hazard for what had traditionally been considered safe adjuvants in pesticides. Next slide, please. There are many subtle differences that might affect the risk assessment from all parts of the biochemistry of the double-stranded RNA reactions. In this slide, what I'm showing you is an experiment in which RNA silencing was induced by ingestion. So the target animal ate the double-stranded RNA. The double-stranded RNA was provided to them encased in a bacterium. So the bacteria were engineered to produce the double-stranded RNA. That's the top. And on the bottom, it was chemically synthesized double-stranded RNA, but in all respects known, that double-stranded RNA was chemically identical to the RNA in the bacterium. And you can see that the relative silencing of the target gene, and I'm highlighting here the VATPase gene, the relative effectiveness of silencing was different just depending upon how the animal ate the double-stranded RNA. Now, this may not be a problem if it's the intended target gene in your intended organism, but it may be a problem if what you're trying to do is determine whether this is a hazard to an organism you didn't want to be exposed to the double-stranded RNA. So if you use a delivery mechanism that's biased against causing an observable effect because of the limits of detection of your experiment in your unintended exposed organism, you may miss this toxic effect. Next slide, please. And it's also possible that you will miss an effect even when you have a set list of non-target organisms you're comparing to. So on the list, on the left is an experiment that used a pool of 50 different double-stranded RNAs. And 6 of those 50, which were intended to kill the Western corn rootworm, only 6 of those 50 actually cause greater than 75% mortality. So just as we can't always predict what genes they will affect when we don't want them to, we are still not all that good at predicting when they should have an effect too. 46 out of those 50 caused an effect in a non-target organism, this beetle that they didn't want to kill. And they weren't the same double-stranded RNAs. So it could be that those 6 that they choose for the corn rootworm still have an effect on the beetle, but an effect that's not lethal. So it could be translation inhibition, or it could be transcriptional silencing. And the phenotype is not as easy to see as one that causes a high degree of mortality. So if the limit of detection isn't tuned to that possibility, it could be lost in that environmental risk assessment. On the right is a nifty little paper that looked at the movement of the double-stranded RNA through the food chain. So in this case, what they had was a plant whose roots were soaking in a water solution with a double-stranded RNA active ingredient. The double-stranded RNA flowed up through the plant, and the plant hopper pest was allowed to feed on the plant. Now, when it did so, there was a strong RNAi effect in the plant hopper. They then added a third layer, which is the wolf spider that eats plant hoppers. And what they found was when the wolf spider ate the plant hopper that had eaten the plant, genes in the wolf spider were silenced, but only in tissues around the stomach. So it was limited to the stomach tissues, causing possibly a less obvious phenotypic effect on the wolf spider. But interestingly, if the wolf spider drank little water droplets called guttation water that the plant sweats out, then the entire spider was silenced. So depending on the pathway of exposure, there could be significant differences to non-target organisms and unintended effects in them. Next slide, please. So because of the way these pesticides are going to be delivered in the environment generally in mechanized form, what I'm suggesting here in the center of this slide, all our different types of exposures will be relevant: contact, ingestion, and inhalation, and they may be more or less so depending on what species we're trying to do our risk assessment for. I won't go through all of these boxes, but I just want to reinforce something Magda said, and that is we can use bioinformatics to at least find candidate off-target effects in the first instance, and we should never ignore candidate off-target effects identified by bioinformatics. But bioinformatics is the start of our assessment. They must be supported by empirical evidence, and from whatever country you're in, you'll want to know that those kinds of analyses were done to the appropriate level of limit of detection, through the appropriate exposure pathway for species that are most important to your country. Thanks. Over to Magda.
Yeah, thank you very much, Jack. I mean, lots of information and details. I will go more high level, and I will try to focus on the key conclusions that came out from this OECD work. Next slide, please. So just for those who joined later and maybe they haven't followed the first presentation that I gave, this is a broad set of recommendations related to the environmental and human health risk assessment for these double-stranded RNA-based products. This is a work done by the OECD ad hoc group on RNAi pesticides, and that was conducted under the auspices of OECD Working Party on Pesticides. So here we don't address issues related to the risk assessment of genetically modified crop plants. We focused, as mentioned earlier, and we re-emphasized with Jack, on topically applied double-stranded RNA-based products. So next slide, please. So I will start with our first document on environmental risk assessment. I've explained that that was a priority for the group, and also that was the document that was first released in 2020. So this document contains lots of information and lots of scientific information about the mechanism, the RNA interference mechanism, information about molecular structure of RNA, chemical modifications, assay methods, commercial production, and formulation of these products. Also, you can read about the various types of double-stranded RNA-based end-use products and the application methods. Then there is a special focus on environmental risk assessment, so issues related to non-target effects are discussed, as well as the environmental fate of these products, and also issues for consideration when conducting environmental risk assessment of these products. So if you want to know more about the science, about all these details about the types and products, I would recommend that you go to this first document. Some information is repeated in the second document that we have in human health risk assessment, but it's very limited. So this is more extensive and detailed information if you're interested. And of course, more detailed information about environmental risk assessment. So now we'll share some of these key conclusions. Next slide, please. So the good news is that the expert concluded that government agencies should assess double-stranded RNA molecules developed for topical application to crops for pest control using similar set of considerations to those applied to chemical and biological pesticides. And that's because, maybe next slide, because exposure is important, use pattern and application model are very important to be considered. And these are very different when you compare to the transgenic plants, as we explained. The knowledge that was accumulated from there can be obviously, inform us, but there are some unique aspects that we have to to consider, and these have to do with the use pattern and the application method. And of course, different considerations apply if we are talking about a restricted application of these products in greenhouse, and different if it is outdoor or if it is sprayable or used as a bite product. What we know also, and that was discussed in Jack's presentation, is we have a substantial amount of knowledge that exposure to naked double-stranded RNA is expected to be limited because of low application rates, in comparison to main conventional chemical pesticides. So here we are talking about very targeted approach. So we expect that the application rates will be minimal, very, very low. And also we anticipate a rapid degradation and dissipation of these molecules, but because they are not stable in the environment. But as I would like to stress out, this is knowledge for the naked double-stranded RNA molecules. So next slide. So when we are doing our environmental risk assessment, we need to consider the fate of the double-stranded of the molecules in the environment and the treated crops, and most importantly, the potential to silence genes of other organisms with significant sequence identity with the target gene in the intended pest. So just reiterating again, that's what the experts said, that sequence information alone should not and cannot be used as standalone predictor of off-target effects. Next slide. So bioinformatics can be informative for the selection and prioritization of non-target species for toxicity and effects testing. So basically, they can tell you which species you should select for testing. So here I am explaining basically what I've said at the discussion of the first session, that bioinformatics can be augmented by an empirical approach to introduce these molecules to a range of other organisms, starting with the close relatives. So you can see here, like, or if we are targeting flea beetles, then we're going to the flea beetle predators. And then we're moving outwards to more phylogenetically distant organisms, for example, the beneficial insects like pollinators, bees. Next slide. And also we have this available evidence of long record of safe consumption of naked double-stranded RNA. So through our food, animals through their feed. And we have all these physiological and biochemical barriers that are also listed earlier in the discussion session. So that can limit the uptake and also the distribution of the exogenous RNAs. Next slide. So we are very well equipped in knowledge about barriers to double-stranded RNA uptake in mammals, and also this can apply to other vertebrates. But there is a lack of knowledge in the invertebrate taxa. So we don't know about the barriers that exist in invertebrates to be protected by these molecules. And that's why it makes very important to use, as was emphasized by Jack, bioinformatic information, sequencing data, and then inform us about which species should be tested in the lab to make sure that there is no effect in non-target organisms. So next slide, please. And here is the issue, I mean, that was very well highlighted in the conference, and also is really emphasized in both considerations document, and explained very nicely by Jack, that these products to have an effect, they need to be in a kind of formulation, or they can be modified, chemically modified, to be able to cross all these barriers and being uptaken from the target organisms. So when we know that, obviously some questions are generated. So we need to know, when we regulate these products, how barriers to uptake are proposed to be overcome. So this is something that the generators of these technologies have to share with the regulators, so to understand, what was these formulants, what are these chemical modifications, and how these are barriers to uptake are proposed to be overcome, and what the impact can have in environmental persistence, and the impact to barriers and uptake by non-target organisms. So I cannot stress anymore that here we cannot only rely on the assessment of the active ingredient. We really need to assess the safety of the whole product, because for the conventional pesticides, as you know, in some cases we focus on active ingredients, and we don't actually assess the safety of all of the co-formulants and how they are applied as a product. Only limited testing is done there. So next slide, please. So yes, I mean, where specific product formulation impact barriers, this is like a kind of an alarm, a flag to the regulators to go and test the program, the product on organisms, or test surrogates organisms that can better obviously characterize the potential for hazard. So this testing, of course, we cannot really dictate through our documents. This depends on the legal requirements, and that varies from OECD member countries. But this is what is needed really to have a safe product out in the environment. Next slide, please. So something else that I would like to highlight that came out from the discussion at the conference, and is also very prominent in the consideration documents, is that as I've mentioned earlier in my first talk, we have the tools. So we have, for example, OECD test guidelines to assess safety of chemicals. But the protocols of these test methods, maybe they should be modified. And that's because these products require some longer time to display efficacy. So any evaluation for these products needs to take account for this timeline by extending the study observation period. Additionally, any evaluation of these pesticides should include monitoring of degradation. So we know if this product, if this molecule is stable, and what we measure in terms of effects to organisms is something real or not, because the product was degraded. And of course, there are lots of uncertainties. So if we picked up something in a higher tier, then we may need to know more things, to go to more complex studies related to the growth, development, and reproduction, or to more specialized other non-lethal effects. So there are more, more testing that can be done to reassure the regulator that this is safe product to be released in the environment. And next slide, please. So very quickly moving to the human health risk assessment here, the considerations are pretty much the same about the bioinformatics, about exposure. What I want to highlight it is that for the human health, we are not only concerned about oral exposure, we are also concerned about dermal exposure for workers, but on top also about respiratory exposure and ocular exposure. And here where the studies that they've done in the medical field can help a lot. For example, we know that these molecules are more stable to ocular fluids. So if these products are coming out, or we need to take some mitigation measures, or we need to know more about this potential exposure, especially for workers and bystanders to these products. And of course, if if the uptake is happening, then there are considerations of possible immune system stimulation and RNA machinery saturation. So there is a lot of literature there and some concerns for this issue. Next slide, please. To save time, to have maybe a few questions, I won't go that. I wouldn't stress more, how important it is to assess a full as a product rather than as an active ingredient. So we need to really learn what is known in the medical field where these products were used as pharmaceuticals. And do some specific empirical testing to fully characterize hazard and exposure. Next slide, please. And again, these modifications in the protocols should be considered. Also, extend the time of reading the results, and also we have a great knowledge gap about issues related to development and reproduction. So regulators also may wish to look at this aspect and other also non-health, non-lethal effects. And with that, I think I'll finish my presentation, and I hope we'll cover you, and that's why I didn't reply to some of the questions earlier, because I thought through our presentations we will cover all these.
Thank you, Magda. And there's been rich discussion going on in the chat as you were presenting. Thank you to both you and Jack. I think you have, with your whole presentation today, you have really given a lot more information to people who are either involved in regulation, or maybe academics or researchers or NGOs who might be impacted by these decisions, to start thinking about the complexity and the challenges behind RNAi pesticides, and particularly as people are raising in terms of regulating them in low and middle income countries. So we have 3 minutes left, and we've got the poll on. I'm going to, Jack and Magda, if you want to make any comments. There are a lot of questions coming through, but there's one, there's a joint question, and see if I can find it now, between Joan and Simon. So sort of asking this question about, what specifically information is needed in order to make a regulatory decision, and how do you suggest that the safety and the regulation of this product or these products actually occur? So it's just a few thoughts and comments, since we don't have much time.
Yeah, okay.
Magda,
So I can try. But this is, different regulatory approaches and different countries that apply. So there are also, some countries they have prescriptive data requirements that they need to be addressed, and that's how you can reassure the safety of the products. Others they go into the different approach, then the need to know approach, and I think what we presented here today really helped to formulate these questions, to the regulators, what is the first thing that I need to know. And I think we explained very well that it's important to understand, how this product is uptaking from the target insect, how barriers are crossed, and then start to structure the assessment. Also, the exposure considerations are very important. So that can also direct and or lead to particular questions, how it can be assessed. But I think we gave a flavor, but it's like different from country to country. So I cannot really say. In our last document, I think the US approach is summarized. As we've said, other countries, they haven't gained any experience yet. So this is something really new. So we'll see in the near future if more tools are needed, actually.
Thank you. Maybe Jack, just before you come in, so the FAO and WHO of the UN have the joint meeting on pesticide management, which I sat on for many years. And I believe, and we have some people who are participating today who are on that committee. And I believe this is one of the things that is being considered as whether a guidance document needs to be developed then for low and middle income countries. And I think the discussion today really stimulated a lot of thinking about that. Jack, do you want to make any other comments? I see we're a bit over time, but any concluding comments from your side?
I would say that, only say that, though these commercial products are developing apace, the science on gene silencing is still a very young science. And we know very little about it in the vast majority of species that are on Earth. And a good proportion of those are the ones likely to be exposed. So I do think we need to concentrate on case-by-case risk assessments, and they have to be ones that we can truly be confident won't cause, particularly in my view, more subtle sublethal effects that can undermine the environmental productivity, but don't have really powerful phenotypic effects on the individual and therefore are statistically difficult to demonstrate. I think those could be a large proportion of off-target effects, and we need to take that into account for the species that are very important for our own ecosystems.
Thank you. I think that's an important conclusion, which you drew out very well in your presentation. So really the goal today was that we have many people on board who are regulators, and particularly in low and middle income countries. And it's, this is a new concept for some regulators who may be presented with applications in the near future or in the future. And I think you both have done an excellent job in giving a lot of background information on what regulators need to start thinking about and questioning, and the questions that they could ask and the pushback they could give in terms of what kind of information they need to make an informed decision, case by case, as you say, Jack. So again, thank you both very much for the rich information you've given. We will have a digest that comes out. I know quite a few questions may not have been addressed, and some of the polls have highlighted things, but we will address these in the digest that we will then circulate to members. If I could have the next slide, please. Next slide, please. So just very quickly to conclude, as we are going over time, is just to, you will have seen that some people have mentioned that their students in our MCRM program, we have applications are open at the moment. So if you're interested in delving into issues like what we discussed today and other around regulation, please apply for our Professional Masters in Chemicals Risk Management, which is fully online. And then we also have a Masters of Public Health and Environmental Health track, which is actually a face-to-face program. So please, we encourage you to apply for these programs. Next slide, please. I did mention that we will have a digest. There is a recording and we will have the slides made available. Next slide, please. And you are encouraged to go to our website and to be able to download these. We can also put the link in the chat. And then the next slide. So we are, just to say again, thank you very much to our presenters and also for accommodating the time, and to our participants. Thank you for the rich discussion. And I hope this is not the last discussion we have on RNAi pesticides, and that we'll be back again sometime discussing. So thank you again for your time. Our next discussion will be on the 23rd of May. We'll be looking at the illegal pesticide trade. You are all most welcome. I hope you'll sign up, maybe even the presenters. You want to join any of our discussions, you're most welcome. So have a good evening, Jack. Have a good day, Magda, and all the best to everyone. See you next time. Take care. Thanks.
Thank you.
Bye.